Hi everyone, my name is Oguz Selvitopi. I'm from Lawrence Berkeley National Lab, and today I will be talking about our work on parallel protein sequence alignment. The focus of my talk is distributed protein similarity search. These are useful structures for detecting protein families. The proteins in a family share significant sequence similarity along other features such as similar three-dimensional structures and functions. Detection of protein families is useful in assigning functions to unfamiliar family members by the help of the well-known proteins within the same family. One way to discover protein families is through the clustering of protein sequences. We can compare sequences and assign similarity scores to them, and then use that information to cluster the network. The existing tools in this area often hit a bottleneck when they are challenged with the massive amount of data associated with modern isolate genome and metagenome databases. Distributed parallel computation is one way to tackle this huge space. There are several existing software that can be used for this purpose. Although these tools are fast and well optimized for shared memory environments, their shortcomings are there when they are challenged with these huge datasets. The techniques used to optimize these tools often do not fare well in distributed memory settings. Our software related to this work is called PASTIS, in short for protein alignment via sparse matrices. PASTIS constructs similarity graphs from large collection of protein sequences, which in turn can be used by a clustering algorithm to discover protein families. A major novelty of PASTIS is its use of distributed sparse matrices as its underlying data structure. In this direction, we develop custom semirings in sparse matrix computations to enable different types of alignments. Some terminology before further moving on. For the rest of my talk, a protein will be assumed to be a sequence of amino acids. Amino acids are represented with characters from a finite alphabet of size 25. A k-mer is a substring of length k. Alignment refers to finding similar regions in two sequences, and similarity refers to a score of likeness between two sequences. In the end, we want to know whether some proteins are homologous or not. The function of a protein is directly dependent on its three-dimensional structure. However, more than often, a similarity in the primary structure might be sufficient for practical purposes, and that is also what we use in this work. Protein homology search is modeled as a sequence similarity searching problem. We are given a set of protein sequences, and we define a protein similarity graph in which the nodes represent the sequences, the edges represent the strength of similarity between the respective sequences. These edges should only exist when the similarity exceeds a given threshold, or otherwise the graph would be completely connected and it would be infeasible to compute. So we are given p-processing elements, a set of proteins, and some similarity constraints, and we want to compute the similarity network here in parallel as efficiently as possible. Here is our pipeline to form the protein similarity graph. Here at the left we have protein sequences, which consist of bonded amino acids. From a given set of proteins, we select pairs which we want to run alignment on. For alignment, there are several variants, generally categorized under local and global alignment. This will produce us a similarity measure, which we can then use to establish how similar the pair is. Usually, we filter out pairs that do not meet certain criteria in similarity or coverage. This finally leads us to the protein similarity graph. This graph is then clustered to get the protein families, but this is not something I'm going to talk about in this talk. First component is the overlapping sequence identification. A straightforward approach would be to compute pairwise alignment over n sequences. So this would produce us a dense and completely connected graph. This is expensive and unnecessary because not all pairs produce good alignments. In reality, a sequence is usually homologous to a handful of sequences. A common technique in this regard is the seed-based approach, in which we first discover sequence pairs with certain number of common k-mers. Here, two relevant questions are how to represent sequences and k-mers and how to find the overlapping sequence pairs. For the first question, we indicate the existence of a k-mer in a sequence within a sparse matrix, in which the rows represent the sequences, the columns represent the k-mers, and another element at the intersection row I and the column j implies the existence of a k-mer j in sequence I. The amino acid alphabet determines the number of all possible k-mers. For proteins, there are a total of 25 amino acid bases we use, and this makes the size of the k-mer space 25 to the power of k. For the non-zero elements in this matrix here, we store custom information. This may be location, score, etc. The location is more relevant at this point, especially that's especially true for local alignments or seed and extend base aligners. Second question is, how do we find the overlapping sequence pairs now that we represent them with a sparse matrix? The answer to this question is to multiply the sparse matrix with its transpose with overloaded multiply and add operations. So here's an example. Here we have two sequences I and k, and they will be represented by rows in this matrix A and by columns in this matrix A transpose. In the output matrix, the element at location ik will indicate whether these two sequences overlap or not. To compute this output element, we would take the inner product of row I and the column k here, and the multiplication operation becomes something like produce me a pair that contains the location information in both sequences, and add operation becomes concatenate all these pairs. Actually, it is a little bit more complicated than this, as we also store information such as the number of common k-mers and the score of the match. From an evolutionary point of view, matching a base A with another base A and matching base M with M are not the same thing, and one is usually more valuable than the other. These are valuable information which can be utilized to increase the representation quality of the protein similarity graph. There are several observations we can make here. First, the output matrix is symmetric. Another observation is that what happens when there are several overlaps. Consider two identical sequences. The output matrix element in the C matrix would contain all indices in those sequences as pairs. We do not want to hurt performance by storing all of them. So we usually store a fixed number of them. Finally, we did not find similar proteins yet. We just know that they might have something in common. So the next step is actually to test out whether they are really similar by aligning them. For alignment, we rely on an external library called CCAN. So this is an open source C++ library that contains algorithms and data structures to analyze sequences with focus on biological data. It is multi-threaded. It has decent vectorization support. There are several alignment algorithms in it. The two of them we use are full Smith-Waterman and the seed and extend with xtrap, denoted with XD. The Smith-Waterman alignment computes a local alignment starting from the beginning of each sequence. In this case, our seed position is ignored, and the seed is used to mark the two sequences as potentially related and worth aligning. The advantage of Smith-Waterman is that its quality does not depend on the seeds we have found. The X-drop alignment is similar to Smith-Waterman, except that it gives a running count of the alignment score and stops aligning if it drops by an X amount. The advantage of this alignment is that it is faster than the Smith-Waterman alignment. These alignment algorithms are going to produce us some alignment statistics we need, such as the identity, row score, and the coverage, and these are going to be used to form the protein similarity graph. After the pairwise alignment, the sequence pairs that do not meet identity and the coverage criteria are removed. For the similarity of a pair, we require a 30% average nucleotide identity, and for coverage, we require that 70% of the shorter sequence is aligned. After this stage, we have the protein similarity graph. In this graph, the nodes represent the protein sequences, the edges represent the similar sequences, and the weight of an edge signifies the strength of similarity between the respective uh, pair of sequences. PASTIS provides a scalable distributed implementation of this described workflow. The connections found in the protein similarity graph are oblivious to the number of processes used to parallelize PASTIS. It's an important aspect of our tool, as it allows reproducible results. PASTIS relies on two libraries. The first is COMBLAS, and it is used for distributed sparse matrix operations. It supports MPI OpenMP type of hybrid parallelism. The second library, as I also mentioned earlier, is CCAN. It supports shared memory parallelization, and it has vectorization support. PASTIS indirectly makes use of MPI and OpenMP parallelism through these libraries. In addition, PASTIS itself also makes use of hybrid MPI OpenMP type of parallelism in certain components. The input to PASTIS is a set of protein sequences. In FASTA format, we use parallel I/O to read independent chunks of this file here. Each process gets the size of the file and then divides this size evenly by the number of processes. The processes then start communicating sequences to fit into a two-dimensional distribution. So this two-dimensional distribution is imposed by COMBLAS because it makes use of 2D decomposition for sparse matrices. This decomposition dictates the sequence pairs that need to be aligned. But here, as you can see, a particular process may not have the corresponding sequences in its partition. Any remaining sequences it would need has to be fetched from other processes that contain them. In the fetching of remote sequences, a process has the option to either wait until the output matrix is computed, or it can request the full range of sequences it might need immediately. The advantage of this latter approach is that it allows to perform remote sequence fetching in the background while the operations such as seed discovery, the matrix creation, or the multiplication are being performed. Here, this figure illustrates how the implementation of overlapping communication in PASTIS is handled. Immediately after reading the local sequences, each process computes the ranks that it has to request from, as well as the ranks that will request from it. It will then proceed to issue the necessary MPI calls to initiate the remote sequence exchange. After computing the output matrix, an MPI wait tool will guarantee us that each process has received the sequences it needs. Once the output is computed, the non-zero sequence pairs in its lower or upper triangular portion needs to be aligned. If this were done naively, certain processes would sit idle. Alternatively, one could redistribute the pairs in the upper triangular portion among all processes, but that would incur additional communication cost. We can avoid both of this and move computation to data for free since this matrix is symmetric. This is shown in figure at right here. We see that all pairs in the upper triangular portion of this matrix at right are equivalent to the collection of pairs in individual upper triangular portions of each process block. The way we did overlap detection results in exact k-mer matches. However, exact matches pose a strict constraint and they may cause low recall. In bioinformatics, the Blossom matrix is a substitution matrix used for sequence alignment of proteins. These are used to quantify the chance of a certain amino acid being substituted by another during the evolution. For example, here if we match the k-mer ISV to ISV with a score of 12, this would be an exact match. However, there are two other matches with higher scores. These matches are not exact, but still they do have a higher score than exact match. Here, for example, CFH has one substitute and it has a match score of 20, and CFN has two substitutes and it has a match score of 13. We define a fixed size neighborhood for each k-mer, and we adopt the notion of evolution for k-mers. by producing a set of substitute k-mers for any given k-mer. Our algorithm chooses the one with the highest chance to appear in place of the original k-mer. Here we restrict ourselves to computing m nearest such substitutes for any original k-mer present in a sequence. Here observe that the m nearest k-mers are not restricted to single substitutions. Those neighbors of a given k-mer can be multiple hops away in terms of the edit distance. So how does this fit into our pipeline? We introduce a new matrix to capture this substitution information. The rows and the columns in this matrix represent the k-mers, and another at location I at the intersection of row and column I indicates that k-mer j is within the neighborhood, m neighborhood of k-mer I. At each non-zero we store custom information. That will be the substitution cost. For the exact k-mers, we multiply A with A transpose to find the overlapping sequences. When the substitute k-mers are involved, we first multiply A with the substitute matrix, and then to discover the overlaps, we multiply it with its transpose. We use a custom semiring here. This semiring is different from the one that is used for the exact k-mers, as it also accounts for the substitution costs. We conduct our experiments under two categories of parallel performance and sensitivity. For parallel performance, we use subsets of a dataset that contains 313 million sequences. For sensitivity analysis, we use a dataset that is manually created. This dataset contains 77K proteins and 5,000 protein families. Our competitors here are two state-of-the-art software, MMSIG2 and LAST. These are originally shared memory parallel software, but MMSIG2 has also an MPI mode. We conduct our experiments on NERSC query system. That's a Cray machine. It consists of two partitions, the Haswell partition and the KNL partition that contains nice landing CPUs. We use Haswell nodes in our comparisons against MMSIG2 and LAST, while we use the KNL partition in analyzing the scalability of PASTIS. For the comparison, we use two datasets with 500,000 and 1 million sequences. We vary the number of nodes from 1 to 256, increasing it by a factor of 4. Additionally, we evaluate a number of different options for these tools. For PASTIS, we test out Smith-Waterman and X-drop alignments. These are indicated with the suffixes SW and XD here. For MMSIG2, we test out three different sensitivity settings, indicated with low, default, and high. A smaller sensitivity value should result in a faster execution for MMSIG2. For the software LAST, we use the defaults. Here, in this plot, both axes are in log scale with a base of 4. Both of these figures show that the PASTIS is slower than MMSIG2 for small node counts, but due to its better scalability, it is able to close the performance gap rather quickly. So, PASTIS with X-drop runs faster than MMSIG2 in both datasets, starting around 16 nodes. And PASTIS often scales favorably. This figure shows the strong scaling performance of PASTIS. Regarding the figure, using the exact k-mers exhibit better scalability than using the substitute k-mers up to 2K nodes. The substitute k-mers exhibit similar scalability among themselves, with an increase in runtime as the number of substitutes increases. The substitute k-mers implementation requires formation of the substitute matrix F, and an additional multiplication and the symmetrization of the similarity matrix. These components seem to be less scalable compared to other components. This figure shows the percentage of time spent in various components of PASTIS. Here we exclude alignment from our analysis. There are different components, and the components that are specific to substitutes are the formation of S, the substitute matrix, the multiplication of the substitute matrix with the matrix A, and the symmetrization. For the exact cases, the most computational dominant expensive component is the multiplication, whereas for the substitute, the multiplication and the formation of S dominate the computation. The formation of the matrices A and S often takes less time than the multiplications at smaller node counts. However, with increasing number of nodes, the percentage of time spent in the multiplication increases as opposed to that of matrix formation, which indicates that the multiplication is less scalable. These two figures illustrate how different components of PASTIS scale. The figure on the left is for PASTIS with exact k-mers and 2.5 million sequences. The one on the right is for the same dataset with 25 substitute k-mers. In both plots, the bottleneck for scalability seems to be the multiplication operations. Other components either take too short time or they scale relatively better. Finally, this figure illustrates the precision and recall of PASTIS MMSIG2 and LAS on the scope dataset. The annotation text over PASTIS variants indicate the number of substitutes utilized in the corresponding setting. The sensitivity parameters for MMSIG2 variants here range from left to right as low, default, and high. We also have a similar sensitivity setting for the software LAST. In our evaluation here, we took the similarity graphs produced by PASTIS, MMSIG2, and LAST, and then these graphs are clustered to discover possible protein families. To determine the edge weights in this graph, we use the average nucleotide identity similarity score. For PASTIS, this figure shows that one can use different number of substitutes to adjust the sensitivity, where increasing the number of substitutes increases the recall, at the expense of precision, obviously. And the takeaway from this figure is that PASTIS is able to achieve competitive sensitivity with other established tools in this area. So with the expressive power of sparse matrix operations and those operations' strong parallel performance, our approach enables construction of huge similarity networks on thousands of nodes in parallel. The substitutes in PASTIS facilitate better recall and enables a control over sensitivity. With several optimizations, our software PASTIS illustrates how linear algebra can provide a flexible medium to tackle problems in computational biology that deal with huge datasets. As future work, we first plan to address the issues related to memory requirements of PASTIS, as it occasionally runs out of memory at small node counts. A direction in this regard is the partial formation of the output matrix. We also plan to conduct a performance analysis of a NAS pipeline that involves also the clustering. Thank you for your attention. If you have any questions, I would be glad to answer them.